Hello, and welcome to today's live webinar, Using Data to Drive Automated Screening and Effective Reporting. I'm Robert Castellanos of LabRoots, and I'll be moderating for today's event. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It is also sponsored by Beckman Coulter. At Beckman Coulter, we are dedicated to advancing and optimizing the laboratory. For more than 80 years, we have been a trusted partner for laboratory professionals, helping to advance scientific research and patient care. We have a vital role. Our focus on innovation, reliability, and efficiency has led us to become the partner of choice for clinical research and industrial customers around the globe. For more information, please visit beckman.com. So let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them in the Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems hearing this presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at the lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the C button located in the bottom left-hand corner of your web page and follow the process of obtaining your credits. I would like to now introduce you to today's speakers, Tim Sherrill and Dr. Mike Kowalski. Tim is a SAS systems engineer in the Integrated Solutions Group of Beckman Coulter Life Sciences. He received his degree in computer science from Rolls-Holman Institute of Technology. Tim has spent his career with Beckman Coulter in the areas of software research and development, custom engineering services, field project management, and life science automation. His projects have focused in the areas of automated data processing and last year a new version of Beckman Coulter's data management project, DART. Dr. Mike Kowalski is a staff application scientist in the Sample Preparation and Applied Markets Group of Beckman Coulter's Life Sciences. He received his PhD in microbiology and molecular genetics from Harvard University. Prior to joining Beckman, Mike was a postdoctoral fellow at Novartis Institute for Biomedical Research, where he used automation to screen for novel regulatories of stem cells, pluripotency, and differentiation. Since joining Beckman, Mike has developed automated applications in the areas of cell culture, cellular analysis, and mass spectrometry. I would like to now hand it over to today's presenters.
Well, welcome. We're very excited to be here giving you this webinar. And today we'd like to talk about using data to drive automated screening and effective reporting. Today we're going to take a look at how to create a data management plan and the components that go into that. And then Mike's going to cover information about how he put together a data management plan for a specific screen for cancer regulators. As we look at a data management plan, this is a little different than a data plan on your phone. This is something where you want to be looking at planning out how your data flows through a system. And data means a lot of different things. There's different layers, different types, there's different ways of looking at things, and it means a lot of different things to different people. So today we're going to take a look at how we can organize data in a way that is more meaningful and that can drive results through your assay. Today we'll follow a process of a workflow where you're importing data in the beginning, using data to drive your process, and then look at some analysis. And we'll come back to this workflow several times as we move through the presentation. Let's first look at the types of data in the lab. For instance, there's method and run data, labware properties, and well properties. We'll go through these all in more detail. And then we'll look at a taxonomy of data as it applies to an instrument, such as the Biomech automated pipetter is shown here. This is where data comes into the system and influences it in order to produce results that you can work with. We'll discuss 3 data management tools that we use at Beckman Coulter to perform automated workflows that process both your samples and your data together. And then finally, we'll look at the data workflow cycle: import, process, and analyze.
So we'll be also using, as Tim mentioned, an example of a microRNA screen as a way to show and demonstrate how the data moves through an automated system. A little more background on the screen. The goal was to identify microRNAs that can affect either cancer cell growth or chemosensitivity to chemotherapeutics. The approach we took in brief was to transfect colon carcinoma cell line HCT116 with a microRNA mimic library, and following transfection, we would treat the cells with 5-fluorouracil, a common chemotherapeutic agent for colon carcinoma. We would then count total cells and dead cells as measured by DRAC7 uptake, DRAC7 being a fluorescent reagent that is unable to penetrate normal cells but can penetrate through damaged cell membranes. And we would assay these cells at multiple time points to gain a greater understanding of what's occurring in the system. And we'll go through this in a bit more detail. More on the specific workflow. So the microRNA library, which we purchased from Sigma, arrived in 24 96-well plates to both conserve reagents as well as conserve space on the deck and in the storage areas. We reconsolidated these into 6 384-well plates after rehydrating the libraries. During the actual screen, those library plates were then diluted into media and added with a transfection lipid reagent, in this case Darmafect-1. And this was all occurring in black-walled, clear-bottom 384-well assay plates, again, since we were going to use imaging for our analysis. These reagents were then stamped into replicate plates, so we now have 12 384-well plates, again, all of the assay variety. And cells were then added to each of the 12 plates to begin the transfection process. After 24 hours, we added both 5-fluorouracil to begin the induction of apoptosis, as well as the DRAC7 reagent to look at the immediate staining following transfection with the microRNAs. We then did an immediate read at 2 different wavelengths using the SpectraMax i3X with Minimax imaging cytometer, and we looked on bright field to do a total cell count and looked at 731 nanometers to look at DRAC7 uptake. And again, measuring at day 1, we were identifying microRNAs whose overexpression was sufficient to induce cytotoxicity in cells. We then reanalyzed the cells 24 hours later, so now the cells have been in the presence of 5-fluorouracil for 24 hours, and here we would hope to identify microRNAs that could enhance the toxicity of 5-fluorouracil. We then ran the analysis again at day 3, and now most of the cells will have undergone apoptosis due to 5-fluorouracil, but we might be able to identify microRNAs that could actually inhibit this process, and those microRNAs could then be future targets for chemotherapeutic intervention. So to give you an idea of what this all translates to from a data perspective, obviously we started with 6 384-well library plates, 384 wells each, with 2 replicate assay plates for each well, 2 wavelengths that we were analyzing at 3 different time points for a grand total of 27,648 data points, simply coming off the analyzer, and that needs to be mapped back to our library. And this doesn't take into account other pieces of data that we'll be describing as we go through the talk. This is an image of the system that we use to actually execute the screen. Again, it's centered around the Biomech FXP, which Tim mentioned earlier. This has a multi-channel and a span-8 head, allowing us a lot of flexibility for our liquid handling. And since this is a cell culture application, we enclosed it in a HEPA-filtered enclosure to make sure that the cells were maintained in a sterile state during manipulation. Empty plates and tips were brought from the storage component on the left-hand side, and as cells were treated, they were then stored in the incubator on the right-hand side. The plates were brought in and off the system using a shuttle and would pass through barcode readers to identify the plates as they moved on and off. The Minimax, shown on the left-hand side, was also integrated with the system, and plates were then placed on the Minimax for analysis using the BERT robotic transport shown on the left. Again, this is all, you can imagine, a very potentially complex system, but the goal here is to integrate as much as possible so that it minimizes the amount of user interaction to help minimize the number of mistakes that can be made in your sample processing. And the same is true for the data. We want to have that be as integrated and as seamless as possible so there's less chance to introduce errors.
Thanks for going through all of that, Mike. I really do appreciate that there are people like you that know all the science stuff so that I can stay in my comfort zone of the data. But really, as we look at automation and as I talk to customers who are looking to automate their workflow, we need to automate both the sample processing and automate that assay that you've been working hard on. But if you haven't brought the data through that process, then you've only done half the job. It's really the data that comes out of the system that's important. So let's take a look at the different types of data that we'll be dealing with. So I'll walk through a hierarchy of 3 different levels. So first of all, at the highest level, we have method data. This is something that's global to the run or to the assay that's being performed. It's not specific or varying from one plate to the next or from well to well, but it is specific to what's happening on this day with this particular assay.
So some examples of method data from the screen could be such things as the user. If we have a login for the biomech, you can tell who's been using the system to run which assays. What's the name of the method? Am I currently running my transfection protocol or one of my analysis protocols? What's the start time? So I mentioned it was 24 hours later after transfection that I wanted to analyze, but was it 24 hours or was it really 21 or 27 or somewhere thereabouts? Was there anything noteworthy that is worth making notes on for a given process? And although this particular screen only had 12 total plates, you can imagine a much larger screen, such as one that would have 100 plates. You might want to process them in 10 batches of 10, so you could imagine something like a batch number, which would apply to all the plates that went through a given run.
One layer further down, we look at labware data. This would be something that's specific to one piece of labware that's being used in this automated run. You can have multiple layers or levels that are assigned to that piece of labware, but it will be distinct from one piece of labware to the next.
Examples from the screen, the main one would be the barcode. This is really what was the unique identifier for each plate and one that would confirm the plate's identity as it was brought out of storage or out of the incubator. But you can also imagine other runs where you might just have other identifiers such as a plate name, which you could give it, or a given labware type. Is it a black-walled clear bottom plate or is it round or flat-bottomed? As well as information such as the storage position of the plate. Where is it currently located in the system? Is it in the incubator? Is it being analyzed currently? And how long has a given plate been in or out of the incubator? You can certainly imagine that if I forgot to put a box of tips in the plate would have been brought out and then realized that there were no tips present, so it would be waiting for me to fix the system. This information could be tracked, so if I did see something abnormal for that given plate, we would have that type of information knowing that maybe it was out of the incubator for a longer time than the others.
The lowest level that we'll go into today is talking about well data. This is something that will vary from one well to the next. Often it may be called an array of information, but it's about the wells on one microplate or one rack of tubes. And again, multiple values per well would be allowed, as there can be many different characteristics that are applied to an individual well.
Some examples from the screen, obviously the main ones of interest would be the microRNA that is present in a given well or in a set of given replicate wells, as well as the sequence of that microRNA. Additional information which might be all the same across the plate, but we chose to label it as well information because it could vary, include such things as the transfection lipid, the cell line that we're using, the compound such as 5-fluorouracil that we're treating with, and the volume present in each well of each plate. Finally, when it comes to the analytical data, obviously the main results we're interested in are our total cell counts at all the different time points and our DRAC7 counts at different time points.
So now we've covered the first third of our presentation, looking at the different hierarchy of data with method, labware, and well data. So if you do have any questions, take a moment to go ahead and enter that question, and we'll move on to talk about 3 data management tools.
So the first tool is Sammy TX. This is one of our softwares that we use when it comes to processing batches of plates. Essentially, you want to run the same process over and over on multiple plates. Sammy is great at creating these types of workflows that will process a plate through a variety of different treatments. And this helps you manage those integrated resources that I described earlier. So again, it won't necessarily take a plate out of the integrated incubator until it's ready to be processed on the biomech. This is also great for scheduling actions, as you can see in the Gantt chart down below. Basically, Sammy is trying to process samples as most efficiently as possible, but it needs to know where all the labware is at different times and how it can be processed most efficiently.
So the job that Sammy TX does from a data perspective is it attaches data to source plates. It copies information from a source plate to a destination plate, such as when pipetting occurs. It collects analytical data from readers or other instruments that produce data, such as a 2D barcode reader, and then attaches that to plates as well data. And then it also can be driven through information that's on labware or at the well level to affect the direction that automation takes. Some hit picking or some if statement type functionality.
The next tool is DART, our data acquisition and reporting tool. And what this is an excellent data repository. And we'll describe a lot more about how this works as the talk goes on. But what's nice is that it works either with Biomech or with Sami or both softwares. And one of the nice features that I really enjoyed using was the fact that it allows me to view data as it's coming off live, but I can view it remotely. So I don't necessarily have to be at the system to watch data as it's produced. I can be doing this from my laptop back at my desk.
The job that DART does from a data perspective is collecting information from the Sammy or Biomech method. As soon as that information is learned in the Sammy or Biomech software, it's stored away in the data repository. If running multiple methods on multiple days or on multiple instruments, DART is able to retrieve that plate data. So nothing needs to be re-imported. Just simply reading the barcode and loading the data returns any information known about that plate. So now you can add additional layers of information or make decisions based on information that's in the repository. And then finally, information isn't trapped in your database. There's a flexible web and Excel-based reporting tool that allows you to format the reports as you wish or to drill down and to explore the information through the web. Our final piece of data management tool is the Sammy process management software, and what this can do is link Sammy methods over days and weeks. And this is really key for those cell culture type of applications that we're describing today. Obviously, my screen took place over the course of 3 active days, not including the reformatting steps that were done earlier. But if you can certainly imagine workflows that might take 8 to 14 days, or you can imagine a continuous cell culture type of process, which could go on for months. And Sammy can schedule the activities on a calendar so you can see what resources are needed, as well as what actions are going to take place on a given day. If all you're doing is running a simple set of methods, Sammy process management does not bring a lot of value. But if you're running multiple processes with multiple batches of plates, it helps you to make sure you're running the right method on the right plates at the right time. It also allows you to look at when your system is available or when it has been reserved so that you can plan out, am I able to start this on next Wednesday because the following Friday we actually need to take the system down for maintenance. It can help you to make those types of decisions. So there we moved quickly through 3 data management tools. Once again, these are the tools that Beckman Coulter uses for data management. There are other tools on the market available. What we really want to talk to you about is how to make a data management plan that sets you up for success. So we'll move to the final element of our discussion today, talking about the data workflow. There are really 3 purposes that data can serve in an assay. And one is around identification. The next is around process information, that either information coming in that drives the process or information that is discovered as part of the process, such as reading a barcode. And then finally there's analysis. This is really the goal and the purpose of running the assay. It's really not about moving liquid or taking it to a reader. It's really about understanding the results of that read and being able to draw conclusions from it. So here we have a data management plan workflow. It goes through an import process, through an import step, a processing step, and an analysis step. And then once you've drawn conclusions about your results, then almost always there's a follow-on step, another process step begins. And that might be downstream in another lab, or it might be your self-repeating results, confirming results, or retesting items. But this is the data workflow. So the data workflow on our screen, Mike will go into here.
Yep, so sticking with the idea of focusing on importing data, the main piece of data we needed to import was our library information. So we did receive a CSV file from the manufacturer at Sigma, basically indicating what microRNAs were present in which wells of the original 24 or 96 well plates, as well as sequence information, accession numbers, etc. And so we were able to within the Biomech software, import that information and apply it to those original 24 well plates and link it to those barcodes. when we actually executed the liquid handling steps for the reformatting into the 384 well plates, that additional information was transferred to those new plates, the new 384 well library plates, and that information was then linked to the barcodes of those 384 well plates and stored in the Dart repository.
Here's how this would look like using our tool. So in the background, we see the CSV file that was provided by Sigma, and we attach it to the source plate. In this case, this is the library plate, which is in the lower left of that of the workflow that you see, a snapshot of a Sammy method. This information is brought into the system, and then we can choose which columns of information are attached to that plate, and that those columns can be brought in as either labware data or as well data.
So basically what this looks like from the Dart data repository tool is as follows. Following this initial reformatting, we have shown in red the data that we've imported. Again, as Tim mentioned, we only chose to import the microRNA ID and microRNA sequence, and that is now the well level data that's linked to the barcode of the 384 well plates. But shown here in green is additional process information that came out of that initial run, such as we mentioned before, the method name, start and finish times at the run level. At the plate or labware level, we have the barcode, which I've already mentioned, as well as the name and storage position of those library plates. And at the well level, we actually have the updated volume information for each well in the new 384 well library plates. So moving forward from that, now that we have this information, there's additional processes that the library plates obviously undergo. So when the library is brought back onto the system and the barcode is scanned, that additional information of the microRNA is accessed from the Dart repository, and as we transfer the libraries to the assay plates, once again, that data is taken forward throughout the process, as well as then brought back from the Dart repository during the replicate stamping. So that information is now present on our 12 assay plates. And when cells are added, that additional information gets added as well.
So we first brought the information onto the plate. That was the import step. And so now we need to bring that information through the system. Now the information was all attached to the library plate, but we need it on the assay plate. There's really two ways to work with correlation of data. One is to get your results and then work backwards from the well transfers and go to the beginning of what was transferred through all of these plates. But a much simpler way to go is to first bring the information onto the library plate. The target is to put it on the assay plate so that we can have columns side by side, the analysis with the microRNA ID. And how does this get accomplished? It happens in a transfer node. The Biomech and Sammy software, when we transfer sample, we also transfer data. The information about the sample from the library plate, even though it's being reformatted from 96 to 384, even though it's being moved into replicate plates, that information all will get transferred into that destination. Later on, when we collect the analytical information, we'll now be able to put those two pieces side by side.
So as Tim mentioned, this gives you an idea of what the repository would have in it now. Again, we have brought that information that we originally imported, still shown in red. That has been brought forward to the new assay plates and linked to the barcode, as well as updated information from the process, such as what lipid was added and what cell line was added, and as well as having new updated volumes for all the library and assay plates. So the next step in the workflow is, again, to first add the compounds, the 5-fluorouracil and the DRAX7, and then we begin the acquisition of our analysis data. So again, we get the minimax reads at 2 different wavelengths at 3 different time points on day 1, day 2, and day 3. So how do we actually attach that data to the labware? So Tim will explain this in our Sammy method.
So here's a simple Sammy method. It's just linear. It's take the plate out, get it to the reader, and then store it back away. In this case, we have 2 plates being run through simultaneously. That was just a decision on how to most efficiently use the system. And there are 2 different reader nodes on each line. The first one is doing your cell count and attaching that to the plate. The second read is the DRAX7 count, and this is then run at 3 different time points. So this method gets run 3 times and produces, in this case, 6 different sets of data.
So at the completion of the screen, what our data repository should look like is basically very similar to what we've seen previously, but now shown in purple is the analytical data that has been added. As Tim mentioned, we have 3 different time points with 2 different wavelengths each. And what you can see is one of the benefits is that we now have that, because the data has been brought forward, we now have all of the information we need to do a direct comparison. There's no need to backtrack and try and figure out what microRNA corresponds to which analytical result. All of that information has been brought forward, so it's all linked to the assay plate by the barcode. In addition, we now have also added some more process information, such as the compound that's been added, in this case 5-fluorouracil. And once again, all of that information is present in one location, so we do not have to do any additional backtracking.
The real value of this is that we have now brought the microRNA IDs, the sequence, and put it right next to the information on that we achieved from the reader. Looking at the process data, this is a very important piece of making a data management plan. Just as you would look at, what do I need in my system to pipette from one plate to another? Do I need a dispenser? Do I need a washer? Do I need some sort of heating or cooling on my system? The same thing you should be looking for is to make sure that your data management needs are being addressed through software. So a couple of things that the Beckman-Coulter software can offer are, these are some of the verbs that are available in the system. Being able to create information from a text file or importing it, being able to copy or delete, rename information, being able to do math on a data set, to do division, addition, subtract out a background, do all those sorts of things. Load information from a previous run. All of these are verbs that are accomplished through the Beckman-Coulter software, and none of them require any sort of database knowledge or SQL queries. None of them have any sort of particular data container. It's just a natural workflow that just integrates into your method. And this is the most important element of a data management plan, is to ensure that as you move your sample, that your data moves along and that any new information is attached to that plate or to the well in an appropriate way. So DART is the key that brings all of these pieces together. So the information can come in from a handheld scanner or from an import. It can be incorporated from one or more different automated systems. It can also be annotated or viewed from remotely. Any device that can see the DART database, so this would be something probably limited to inside your enterprise environment, can now browse that DART data and view it. Likewise, any data producers, such as a microplate reader or a 2D barcode scanner, can also publish information into this system. DART is what is able to connect all this information together, and then once again, if your data is trapped in a system or if it's trapped in CSV files on disk, if it requires copying and pasting in order to do anything, any sort of management with it, then your software system really isn't doing all the jobs that it could be. And so you want a system that not only moves that information through the system, but then gives you a good way to view it as you perform your analysis. In the case of DART, we have 2 different tools that can operate on that data. So the first is a query builder, and this allows you to choose the columns that you want to see in a report. On the left-hand side are all the different values, the run characteristics, the labware properties, and the well values that have been captured as part of an automated run. And then on the right-hand side, you're just selecting which columns you want to see. Now, the real value in DART is that it's going to collect all of the different values and store them so that if you ever wish to come back and look at new information, it's available. So when you're performing your report, you don't need to clutter it with information that you might need later. You just put the things that you want to look at, and you have a nice, concise view of that information in Excel. And even better, once you're in Excel, you have a live link back to that database. So as you perform new runs or have more daily operation, you can have cascading charts or other sheets that evaluate from this from this named range of data. And as you add new information, as you run more information, as you run more samples through your system, then more information is available through the spreadsheet. So rather than producing a new spreadsheet every day, you're able to just click the refresh button. All that information comes into the system, all of your charts and graphs update, and you can use the same tool one day to the next, get that spreadsheet optimized for just the way that you like to look at it. So the other view of this is through the web. This is an example from a laptop computer that could be viewing this from another room. In the case of Mike's system, it's tucked away in an applications lab and in a clean room, and so he gowns up to go in there. So if he'd like to take a look at how things are going, he could use this from his desk out in cubicle land and be able to drill down into it. As we just kind of look at some different pieces here, we can look at the different pieces of data that we talked about earlier. Here's our run data. Here's where we can drill down into plate properties. And then the large view there, the plate map, shows the different layers of well data. And there's a drop down where you can switch between those, or you can see them all in columns to look at things side by side. And that allows for some real drill down. Was this useful to you, Mike, as you were going through?
It was. There were a couple times, for example, during just doing some of the initial optimization experiments, rather than having to export things to Excel and really kind of compare things there, I was able to just look immediately in the data browser to do an initial correlation between results and microRNA in my library. And also during the actual runs, because we were running 12 different reads on a given day, this would extend over about a 6.5-hour time period. So to have to sit there and watch for 6.5 hours to make sure plates come out, are read 2 different times, certainly helps to have the physical integration of the instrument, so I didn't have to sit there and replace plates every 15 or 30 minutes, but it also helps to be able to remotely view the data to make sure that everything's running smoothly. So what was the actual outcome of the screen? Now that we've hopefully shown you how data was processed throughout the workflow and how we were able to get everything out into an Excel format, I was then able to look at my results, and the way I chose to do my analysis was to calculate a percent toxicity in each well. And I achieved this by just dividing the dead cell counts by the total cell counts. And rather than just looking at the DRAC7 counts by themselves, I thought this gave me a better opportunity to possibly normalize wells where due to cell death you may have less total cells, so that may seem like an underrepresented DRAC7 count. But by doing a percent toxicity, that would hopefully be identified. Shown here is just kind of 2 wells from a plate that was 24 hours after transfection. So again, in this case, it would be prior to 5-fluorouracil or simultaneous with 5-fluorouracil addition. So the effects here should only be due to microRNA transfection. Shown in row A at the top is what was one of the wells that achieved the plate mean, so basically the average response I saw on that plate overall. Shown on the left is the bright field image that allowed us to count total cells. And on the right, although it may be a little tough to see, you hopefully can see some red dots that correspond to our DRAC7 staining. And shown below in row B, you'll hopefully notice there are significantly less cells, or the cells appear to be rounded up in the bright field image, and significantly more red staining in the DRAC7 analysis. And by calculating percent toxicity, this corresponded to about a 3-fold increase above the average level of cytotoxicity I saw up in row A. So this would basically be what a hit looks like at this given time point. Shown here is a single plate out of the 12 assay plates we ran, and what we were looking for was, again, basically trying to find some hits that would be considered outliers compared to the average response of transfection conditions. Shown here is one plate, the 320 non-control wells from a 48-hour post-transfection time point, so 24 hours after 5-fluorouracil treatment. And what you can see is that on average, the plate averaged out to about 17% or 18% toxicity for the well. And what we were looking for hits was to find some wells that had more than 2 standard deviations of a response away from the plate mean. So for this, that meant above 34% toxicity or below 2% toxicity. And for it to be considered hit, it had to be present in both replicates. So shown here, there's about 14 wells that you can see for this particular plate that was above the 34% toxicity 2 sigma threshold. But the ones that are marked in red are those 9 hits that actually were present in both replicate wells for these conditions. So what did it all come out to? Well, out of our 1,920 microRNAs that we tested, we came out with about 50 hits for a hit rate of about 2.6%. These hits really broke down into about half of them inducing rapid toxicity unto themselves. So overexpressing the microRNA of 26 microRNAs was sufficient to induce significant toxicity by itself. The remaining 24 are either those microRNAs that can enhance 5-fluorouracil toxicity, or are possibly just slowly inducing toxicity and needed 48 or 72 hours to actually show their effect. But what's also noteworthy is that we did not find any significant inhibitors of toxicity, and that's not really unexpected when we saw that, again, we would have to be achieving below, for example, 2% toxicity at the 48-hour time point. So possibly just due to a result of our stringent conditions, we did not find any microRNAs that seem to inhibit 5-fluorouracil-induced toxicity. Looking at the screens at a bit more granularity, of those 50 hits that we identified, 24 of them seem to have some known functions based on literature. Of those 24, 22 of them were shown to have a role as tumor suppressors. And this actually makes a lot of sense. This is what you would expect your phenotype to be if you overexpressed a microRNA that had a tumor suppressor role. You would expect to see increased toxicity in a cancer cell line. So 22 of our 24 known hits pretty much fell into what we would expect the outcome to be. We did have 2 microRNAs out of our 24 that were identified as oncogenes. And again, you would think if you overexpress this microRNA alone, this would lead to increased cell growth, and so you would possibly expect to see not something that would come up as an increase in toxicity. However, these 2 were only hits in the presence of 5-fluorouracil, so one could certainly expect that driving cell growth in the presence of a chemotherapeutic that is targeting rapidly dividing cells could obviously potentially lead to increased cytotoxicity. So what this really means is that either 22 or likely even 24 of our known 24 microRNAs that have a role in cancer basically came out with the expected outcome. And this really reflects, is kind of a byproduct of having accurate microRNA identification. This really ensures the fact that we did not have some sort of mix-up in the middle of the screen and we were misidentifying our microRNAs. We did not really see any results that seemed to fly in the face of what we would expect. So not only is this basically a nice confirmation about the value of bringing all that data forward on the system, but it actually gives us a lot of confidence in our remaining 26 unknown hits. So these could potentially be legitimate tumor suppressors or, again, other sorts of compounds or other sorts of microRNAs that could enhance 5-fluorouracil toxicity. And so, again, because we have the confidence in our data having been brought forward, we have the confidence to go back and if we want to do some validation and hit validation experiments, we know that we should not have misidentified any of these and possibly be missing some sort of real needle in the haystack novel tumor suppressor due to identifying the wrong microRNA from our screen. So to summarize kind of the screen aspects from the data components, again, as we mentioned, we've successfully managed over 27,000 data points, and that's only counting the reader data. That's not counting the additional process data that we discussed, such as time in and out of the incubator or cell lines added, et cetera. And the benefit is that automating the sample processing, I think we all typically, that's sort of forefront in our mind. We see the value because it takes us away from the bench. But the fact that the automation of the data flow happens seamlessly and without any additional work really is the value is seen when you get your results at the end of the screen. As I mentioned, the data flow, bringing that microRNA data forward onto the system so that it's directly correlating with our analytical results, enabled direct hit mapping. We didn't have to go back and paste the data back in and try and map it back to those microRNA library information. It gives us traceability, which is key sometimes when you get an unexpected result. Certainly when something happens at the bench and I don't get the outcome I'm expecting, my first inclination is to wonder what I've done wrong. But when you have the information all stored in the DART database, you can go back and make sure that everything was done properly and that the results are the legitimate results, and you may need to change your way of thinking. As I mentioned, it gives you the confidence that if you're going to go back for hit validation or you need to confirm any data that you've stored previously, you have the confidence to know that you're going back to the correct microRNA in your library and that whatever hits you identify, whatever hits hold up should be not have introduced any errors in the process. And although we didn't use it for the screen, as we touched on earlier, bringing that data forward can enable data-driven pipetting. For example, at the end of our screen, all we had left were basically dead cells. But if this were a different screen where you were looking for a single clone that was highly expressing a protein of interest, such as an IgG, if your reader data instead of dead cell counts was instead an ELISA read, that information could then be sent back to the system, and that could enable the automation to bring forward clones above a certain threshold, and those clones would then be expanded for further culture on the system.
Yeah, that's a great point, Mike. There's very seldom do these assays get run in isolation. There's almost always something upstream or something downstream, and after this assay is done, the results then feed into the next into the next run. And whether it's in your group or maybe another group, that information usually gets transmitted. So having all that information available in an easy to export format pays a lot of dividends. To begin wrapping up here, if we look at good data design requirements, well, our discussion today really has been about looking at our three different levels of information with our run, our labware, and our well data. We looked at the Sammy EX, the DART, and Sammy process management data manipulation tools, and then we finally looked at our data workflow with importing, processing, and analyzing data. So as you're looking to build a system, a good data management plan needs to do a few things. And this is valuable information when you're looking to plan out how to use data in a laboratory setting. So you need to make sure that you're identifying your compounds or samples. There needs to be some sort of identity. It needs to drive experiment behavior. If you have information about what hits to have or what concentrations to pipette, that needs to automate the liquid handler or other automation in the lab. Information needs to follow the sample when pipetted. The best way to correlate information is to move it forward. So as you move off of your library plate into your assay plate, or if you do serial dilutions or replicate plates, make sure that those identities are moved forward into those subsequent pieces of labware, so that when you do get information, the same plate that has the read value also has the identity that you're trying to track. That makes your analysis much easier because things are already correlated. A good system is going to capture information generated during the process. If you're the one that's copying and pasting results out of one Excel spreadsheet from the MD reader and pasting it into another Excel spreadsheet that has your plate mapping, then you are the data manager. With the Sammy system, we pull that information directly out of the Minimax reader, associate that on the plate, and then we can perform that correlation through DART and in Excel. You need to be able to correlate information. The ID and the results need to be side by side. And then anything that you have learned in the screen, whether it was produced in a report or not, needs to be retained and needs to be something that you've captured and can come back to. There might be some additional insight that as you return to this information, you can extract some sort of additional conclusion from information that you already have. That's where a data tracker can really be valuable. So in successful experiments, recognize workflows, identify the data flows, and you want to make sure that when you're choosing equipment, you also have software that drives it, that can be driven by data, that implements these data workflows and provides reports with the information you need and stores data from every sample every time that it moves.
So with that, we'd just like to thank the folks at Beckman Coulter and also our colleagues at Molecular Devices for the assistance that they gave us, the Integrated Solutions Group for helping us set up the cell culture system, and the group at Molecular Devices for working with us on the i3X with Minimax. And with that, we conclude and thank you for your attention and are happy to take questions at this point.
Well, thank you so very much, Mike and Tim, for your wonderful presentation. I would also like to remind the audience in regards to how to submit some questions by selecting on the green Q&A question tab. All right, we do have a couple of questions that we do have for today. Our first question is, when making a data management plan, should my IT group be involved? And how does this interact with my existing LIMS system?
That's a great question. And I've worked with customer accounts who have both types of scenarios. Sometimes you have, I've talked to some customers, and they say my IT group is so busy. They're really helpful, but they just can't get to me, and so I need to kind of manage my data myself. And so we have tools. We target a laboratory where you're not going to get a lot of assistance from your IT group. Now, on the flip side, we love to work with company IT folks because they can make these tools even more powerful. So we have hooks in there to copy information out of LIMS systems and attach it into our Biomech or Sami software. Likewise, on our reporting side, we have ways that a LIMS system can automatically extract the result data. The same information you're seeing in Excel is available for another database to extract automatically. So really we work either way, and if you have access to a good IT group, count your blessings because not everybody does, and it's wonderful if you have that. And if you don't, we can work with you too, and we can give you some better self-sufficiency.
Well, thank you so very much. Our next question is, what did you mean by talking about pipetting data with sample? And you don't actually pipette data, do you?
Yeah, that's maybe me taking a little bit of liberty here, being figurative. But when the Biomech pipetter puts tips down into a microplate and we aspirate sample, the software behind the scenes is going to look at the well data that is present on that plate. And then we have the different layers of data that are there. Each have a property called pipettable. And if pipettable data is set to true, then the software, in this case virtually, copies that information into the tip, and then any place where that sample is transferred, then that data is also attached to the destination well. So in case of well A1 having sample ID ABC123, let's say we transfer that to an assay plate, and it also goes into well A1. And then maybe we serial dilute, and it goes into B1, C1, and D1. In this case, A1, B1, C1, and D1 of the destination plate will all have the identity ABC123, and any data that gets produced there will be attached back to that sample of origin, and the ID copies along with it. So it's figurative, but I guess the point that is important to me and what we've worked hard to do is that the person writing the method does not need to write a SQL query or do anything specific to copy that information. It happens as a side effect of the liquid transfer.
Thank you so very much. And our next question from the audience is, I spend a lot of time working with CSV files in Excel. Can you describe how your software can make my job easier?
Yeah, we've worked, part of the reason we wanted to do this webinar was to push out some best practice approaches, what we feel are some effective ways of managing data in the lab. And you will never get away from CSV files in the way that laboratories are structured today, but you can use them a lot less. So the way our software works is that you would browse to an input file that you would be using today that identifies the library we're working with today, or the samples that are being processed. Maybe they're being mailed in, maybe they're being fed over to you from another group. But however that information is brought to you, you'll start with a plate or with a rack of tubes, and you'll start with some sort of input file that gives you information about them. And so we'll attach that in the beginning, and that's why we talked about the identity portion of the data workflow. It's at the start of the process. You attach it once. Now, as you drive through the automated system, it will copy that information if you're moving the sample, or maybe you're using information that's attached to the sample to perform some sort of pipetting operation. It could be a threshold, and if the OD value is above your threshold target, then only those wells get copied, and so the automation behaves differently every day because the OD values are different every day, but it automatically selects the ones above your target threshold. It also could be something like normalization, where you're using concentration information to drive a volume that's being transferred into a destination plate. But the idea is that you would use that CSV file, attach it in the beginning, and then you don't have to deal with information, no copying and pasting until you're back in Excel at the end, and that Excel file is being produced as a DART report. that's kind of the gold standard. That's what we would encourage you to strive for, and I think that we've done a good job of removing any of the intermediate CSV files from this type of processing.
All right, thank you so very much. And it looks like we have time for another question here. And how have these tools truly driven ease of managing screening workflows in the lab?
Mike, I've been talking a lot. Do you want to jump on that one?
Sure. I think, again, a lot of it goes back to what Tim's been describing. I think just freeing you up from having to worry about the data aspects during the actual screening itself. As Tim mentioned, we did have an initial CSV file that came from Sigma, and that was the last time I dealt with Excel until I had all of my information at the end of the screen. Being able to just kind of automate that in the background without having to necessarily set up additional steps or additional queries, etc., really enables you to just focus on, obviously, the liquid handling aspects, the part we all think about inherently with automation, but then also whether it's your experimental design or your data analysis, it just frees you up from having to worry about the intermediary steps and allows you to just focus more on the science behind the screens.
All right, well, thank you so very much, and also like to thank the audience for all their questions. Mike and Tim, do you guys have any final comments for today?
I really enjoyed doing this webinar. I'm glad we had folks that we couldn't see on the other end, but Mike, thank you for working with me today. That was really awesome.
Oh, it was my pleasure, and again, it's great to be able to have these tools to use during the screens, and so thanks to you and the Integrated Solutions Group for making my life easier.
All right, well, have a successful time automating your runs.
All right, well, thank you very much, gentlemen. I'd also like to thank our sponsor, Beckman Coulter. This webcast can be viewed on demand through September of 2017. LabRoots will alert you via email when this webcast is available for replay. We encourage you to share that email with your colleagues who may miss today's live event. Until next time, goodbye.